We work with neurotechnology. What's neurotechnology? I think it's the next biotechnology. I think it's the next thing to sweep through and really change how we treat patients. Devices that interface with the brain or the spinal cord to improve function. That's certainly the goal that we work on. You put in examples already like deep brain stimulators. I think the future is bright. So spinal stimulators for pain. I think the future is bright for spinal stimulators for function. And that's really what I'm going to focus on today, as well as just touch on some brain-computer interface work that we've done over the last several years. So with that, we'll divide the talk into two main topics, two goals that we have in the lab. The first goal, shown in the top bullet here, is to directly restore, or what we might call reanimate function. So for example, could we record or decode the intention to move from motor cortex, route that through an implanted device and use it to stimulate the spinal cord or the muscles that are paralyzed below, for example, a cervical spinal cord injury. So we'll show you an example of how that works, both in non-human primates in our research, as well as a couple of clinical trials ongoing around the country. Clinical trials are in muscle stimulation, but we'll talk about why we become fascinated with stimulating the spinal cord below the lesion, both intraspinally, epidurally, and even non-invasively or transcutaneously. And we'll go through examples of each of those, both in animal models and in human trials. And that's what the image on the right here shows, is our non-invasive stimulation approach, which is actually going to multi-site clinical trial this year. It's a collaboration with Dr. Christoph Hofstetter here in your department. And then we'll touch on even some spinal stimulation we're doing for leg function with Dr. Rajiv Saigal, also here in this department. So we can use this picture on the right to kind of schematically break out the talk into three different sections. We'll start, as I said, with brain-computer interfaces for muscle stimulation, show you some initial results in animals there and how they've spawned two clinical trials around the country. Then we'll talk about brain-computer interfaces, not for muscle stimulation, but for spinal stimulation. Show you an example of how we've built a custom circuit that can actually convert brain activity to spinal stimulation in real time, small enough to be implanted under the skin. Could be the beginnings of a new medical device. And talk about some of the advantages we see for stimulating the spinal cord as opposed to stimulating the peripheral nerves and muscles. And then we'll shift from that more direct reanimation focus and spend at least the entire second half of the talk talking about how these neuro devices can induce plasticity, rehabilitation, and long-term recovery. And the key points I want to make here are that we're seeing that these stimulators are having a positive effect that well outlasts the time when the stimulation is actually delivered by up to 6 months, which is as long as we can measure in our experiments. So we're seeing that spinal stimulation especially is leading to improved hand function and walking function that persists for many months beyond the stimulation. So strong evidence for neuroplasticity. We'll talk a little bit about our animal studies that give us some hints at the mechanisms, and then we'll show some highlights of some human trials that show you the functional changes we're observing in people with cervical spinal cord injury. So let's begin here with brain-computer interfaces for muscle stimulation. This is a fun drawing because it was drawn over 10 years ago, well, I guess 12 years ago now, when we first published our paper in monkeys and non-human primates, just demonstrating the simplest version of this concept that a couple of neurons recorded from motor cortex could be used in real time to stimulate a couple of muscles about the wrist. But the artists got excited, they drew this picture, they said, well, this is the system we envision, and it's really satisfying to say that 12 years later now, this system's actually being tested in human subjects. It's almost even more satisfying to say it's being done by other people because it was a massive amount of work, and we'll just highlight that work a little bit and then talk about some of the limitations. So when I was a postdoc here with Eberhard Fetz, Physiology Biophysics, starting in about 2004, we had this idea that we could test Ebb's long demonstrated technology that you could record single neurons from motor cortex. You could operantly condition monkeys to control the activity or the firing rates of those single neurons. But what he hadn't done was connect those neurons to muscle stimulators to control functional electrical stimulation, or FES for short, delivered to paralyzed muscles. And so that was my task as a postdoc in the lab. Turned out the hardest part of that whole experiment was developing the nerve block technique. We didn't want to do a high cervical spinal cord injury in these monkeys. Difficult to maintain the health of the animals. They're precious animals after they've undergone months to years of training. So we developed a reversible nerve block where we would implant epidural catheters into the brachial plexus, and then we could infuse lidocaine or a fast-acting derivative chloroprocaine in order to reversibly paralyze and numb the arm for about 3 to 4 hours during these experiments. Turns out that was the hardest part. It took several years to work out a reliable nerve block method that didn't disturb the animals during the behavioral tasks, so we could test these cells, and then once the nerve was paralyzed, we could stimulate the muscles. Once we made this connection, once we let the animals control in real time their muscle stimulator, they learned it almost instantaneously, achieved very good control. It was a very natural behavior for them, which was good to see. Here's just a quick animation to show the steps of the experiment. We always began by giving the animal biofeedback or visual feedback of the firing rate of those neurons, controlling the smaller white box or cursor on the screen, moving it into the larger box, which was the target. Then we used our neurochip device, or more typically just rack-mounted computer hardware, to convert in real time the activity of 1 or 2 neurons into the stimulation of 1 or 2 antagonist muscles about the wrist. So the monkey's now moving their wrist, which is otherwise paralyzed, to play this simple video game and move the cursor into the box. So it worked fantastically well, but we did notice some limitations, and we'll talk about those limitations in a little bit. And that's what took us, or kept us from taking this to clinical trial. Fortunately, 2 other groups, both in Ohio, actually Chad Boughton pictured here in the upper right. Well, he was at the Ohio State University. He's now at the Feinstein Institute in New York. He worked with Ian Burkhart, who's pictured here, who's a C4 tetraplegic. And Ian volunteered to have a Utah array implanted in motor cortex, a small 96-channel intercortical electrode, recording electrode implanted. They decoded the activity of about 30 to 40 neurons and used that in real time to stimulate the muscles non-invasively through bands of electrodes placed on the forearm. And you can just see them here in the photo. And that allowed Ian to make simple grasping or pinching movements of his hand when he was otherwise paralyzed. And then just a few years later, Bolu Ajiboye at Case Western Reserve implanted this gentleman here, similarly with 1, maybe even 2 Utah rays in this study. And here they used the fully implanted freehand system, which is a set of subcutaneous wires that stimulate the muscles. It's been implanted in about 200 people to help restore grasp and finger movement. It's typically controlled by a shoulder switch on the opposite side for people with C6 level tetraplegia. But here they put that entire system under brain control, including brain control of this mobile arm support, which you can see in black and silver just off the edge of the screen. So I'll pause for a moment and ask you to think, why did they need to use a mobile arm support for this system? The system has electrodes, certainly, that can activate the shoulder and the arm and the hand. What was one of the limitations of muscle stimulation that led them to go to the extra length of putting this mobile arm support under brain control? And feel free to pop an answer in the chat if you think you know the answer, or if you'd like to just raise your hand or unmute and shout it out. I definitely welcome input at this point. Atrophy, one thought from Silas James, thank you very much. Certainly these muscles could have atrophy. There is certainly some atrophy after spinal cord injury. Not the only challenge though. What else do you know about nerve or muscle stimulation that could lead to this need to have an anti-gravity support, something out there that can help support the arm against gravity? Well, good, maybe I'll get to teach you something new. So motor units, motor neurons, and the muscle fibers that they innervate are typically recruited from the smallest, most fatigue-resistant or slow oxidative fibers right through the fastest and most fatigable glycolytic fibers. And we call this Henneman because the way the signals coming down from the brain cross the motor neuron synapse naturally recruits these smaller cell bodies, these smaller fibers first, and then eventually the bigger ones as you have increased descending drive. Unfortunately, functional electrical stimulation and nerve stimulation, whether it's delivered to the muscles or the nerves, recruits these nerves in the absolute opposite order. The first ones you get are the fast fatigable fibers, leads to a difficulty in grading force, but more importantly, very rapid muscle fatigue. And only when you turn that stimulator up to very high currents are you able to recruit the slow oxidative fibers. So that makes FES a challenge. And there's actually been there's been experimental FES systems for walking for over 30 years that allow people to step and stand and climb stairs and sit and turn, but they can only walk about the length of a hospital hallway, and not our amazing health sciences corridor, but like a 50-meter hospital hallway, and then they're completely fatigued. for many hours. And so those systems have never made it into clinical practice because FES has this challenge with recruitment order. And because of those reasons, we've chosen to focus on spinal stimulation, which our colleagues and in our own lab now we're finding recruits motor units at least in a random order, if not in a natural order. So that leads to both fatigue-resistant contractions, but also to more smooth grading of force, the ability to have different outputs. With muscle stimulation, it's often all or none as you activate the muscles because you get those big fibers first. With spinal stimulation, you can get levels in between, levels of force in between. We started our interest in spinal stimulation with intraspinal microstimulation. I'll show you just one or two examples of that. We've since moved to epidural stimulation, which as many of you know is commonly used for chronic pain, implanted over the mid-thoracic or lumbar spine to control chronic back pain. So a little more translatable, and it's being used off-label to treat people with spinal cord injury now. We'll tell you about those studies as well. And then we'll finish up with talking about even non-invasive spinal stimulation, which seems to confer similar benefits. So beginning here with the intraspinal microstimulation, what does that look like? We moved to a rodent model where we could perform a clinically realistic cervical contusion injury, a C4 lateralized contusion injury. And then we tested many sites throughout the cervical enlargement, such as the sites shown here in green. And at all of those sites shown in green, we could evoke graded or proportional extension of the animal's elbow. So this is activating the triceps muscle. The animal's extending its forearm as if it were to be reaching. They're anesthetizing these experiments, but it works in the awake animal as well. And any site in yellow shown here, we can activate flexion of the wrist. And if you look closely, there's a little flexion of the digits as well in this example. So one of the other things we like about spinal stimulation is it activates functional synergies from relatively small numbers of stimulating sites. So we could get a reach and a grasp, for example, from just picking any two of these sites. If we wanted to achieve grasping with the hand with FES, we would need upwards of 8 different electrodes, 8 different muscle and nerve pairs in order to achieve that same functional synergy just for grasping. So we move forward with spinal stimulation to test this concept of a brain-computer interface that could control, instead of muscle stimulation, direct stimulation of the spinal cord. And we demonstrated it both with intraspinal stimulation, but more recently with epidural stimulation, as shown as this picture. So a paddle of electrodes placed on the dorsal surface of the spinal cord, recruits the 1A afferents most likely, and then transsynaptically activates the motor neuron. So taking advantage of that natural recruitment order by moving through the synapse as opposed to directly activating the ventral nerves. Here we made a couple of deliberate decisions, and there's some alphabet soup here that I want to break down for you. So it's important as we talk about moving this to an implanted device. So instead of decoding single neuron action potentials, we used intracortical local field potentials, and you see the power of those in the next animation. High frequency, 200 to 400 hertz frequencies, so high gamma band for Dr. Oachman Jr., who's listening, I'm sure, and just pick the top 8 features from that. And we used the simplest decoder we could find, a canonical correlation analysis, which is just a set of linear models. It's nothing fancy, but the reason we chose it is we could actually run it on a field programmable gate array, or FPGA. So we could run it on a small microchip that could eventually be included in one of these implants. We use that canonical correlation analysis to detect the intention to press the lever, the proportional intention to press the lever. We use that to generate pulses to deliver stimulation to the spinal cord. Animals were motivated by a liquid reward, and they were required to keep their nose right up front and a nose poke sensor, just so their body position was in a constant location relative. Here's what this looks like. The top 8 rows are the power of the 8 most informative local field potentials. Bright colors mean lots of power. It's where we're decoding the activity. The green line is our estimate of the decoder. Does the animal want to press the lever? And if so, by how much? The stimulus trains will be shown here in yellow once the stimulator is enabled, and the movement of the lever is in purple at the bottom. First clip shows the stimulator off, so you'll get a sense of the animal's function after a severe cervical injury. You'll see the brain activity indicating the animal would like to press the lever. The decoder picks that up, but we don't turn on the stimulator until the second clip. So you'll see very little movement of the lever. Current time is here in the middle at the vertical line. As the animal is attempting to press the lever back, you see its brain activity is indicating that, but there's very little movement of the lever other than that generated by the animal's body movement. Now we turn the stimulator on. Again, you'll see that decoded potential. Here's a nice example coming up here. Lots of power in those brain signals, decodes a big lever movement and activates a high stimulus train, which results in the lever being pressed almost all the way back to its limit. So that was exciting. We did most of that with desktop hardware, for full disclosure, but we've always had the goal of building an implanted device that could actually decode intention under the skin. There's all kinds of heat dissipation challenges with this. Most brain-computer interfaces use a state-of-the-art desktop computer or two in order to do real-time decoding. And so we spent quite a bit of time working on a piece of hardware that could be even wirelessly powered. So it doesn't even have a battery and is very, very efficient running under the skin. We haven't yet implanted under the skin, but we have built the device with Josh Smith's lab in electrical and computer engineering and also computer science. Adrian Fairhall helped us quite a bit coming up with the right algorithms that would be as efficient as possible. And so what we show here is about a 3 by 2 centimeter printed circuit board with 2 big chips on it. The Intan does the recording, the amplifying, as well as the stimulating. And then this field programmable gate array, or FPGA, does all of the decoding in real time of these local field potentials. So even though we've gone for a fairly simple BCI, we think it's more clinically translatable because this would easily fit inside one of those titanium cans that makes up, say, a deep brain stimulator or the Activa device that has a little bit of limited real-time processing. All right, so this diagram shows that we bypassed the injury to the spinal cord here and artificially stimulated either within or on the dorsal surface of the spinal cord. But I want to also use this diagram to transition to this concept of plasticity or rehabilitation, which is that operation of circuits like these, at least in the intact nervous system, have been shown to promote plasticity in spared circuits that bypass the lesion. It's been shown in the intact system, has yet to be shown in severe spinal cord injury, but we're starting to see some hints, even with simpler systems, even with systems that just stimulate the spinal cord, but still in an activity-dependent manner, maybe synchronized with the individual's intended movements or practiced movements, we're starting to see evidence of neuroplasticity in spared circuits, such that even if we were to turn the stimulator off or take the device away, people and animals retain a good deal of function that they demonstrate when the stimulator is running. So let's start to talk about that in the last section of the talk. It'll make up about half the time I'm going to spend because I think it's one of the more exciting things we're working on, and we'll see several different examples of spinal interfaces that can induce neuroplasticity, especially when combined with rehabilitation. The first example is in animal models, and it utilizes optogenetics. It's arguably the farthest away from clinical translation, but that's using a viral vector or a transgenic animal that expresses light-sensitive channels in their neurons, such that by shining a blue light, for example, you can activate a subset of neurons or a cell-type specific set of neurons. This particular paper was in collaboration with Paulina Anakiva at MIT. We're also working with Phil Horner, now part of your department formerly, now at Houston, a couple active grants with Phil. First looking at optogenetics alone, but eventually the intriguing possibility of using optogenetics to selectively activate transplanted stem cells. Right? We could make these stem cells optically active before they're grafted and then have selective control over those grafts and maybe over the host tissue around it using a different wavelength of light. So I won't be able to report on that data yet because that's still in progress, but I will tell you what we have done with optogenetics. Even the simple version appears to promote powerful recovery after spinal cord injury. We've mapped the cervical spinal cord similar to the way we mapped it with electrical stimulation. Optogenetics isn't as specific, at least when we use a pan-neuronal promoter like human synapsin in these experiments. We get a lot of movements from throughout the cervical enlargement, movements that look like combinations of withdrawal reflexes, sometimes isolated movements, flexing the bicep here, we call this a curl movement, or moving the shoulder backwards in sort of a rowing movement with latissimus dorsi. Not nearly as specific as intraspinal stimulation, but that appears to be okay because we're not going for reanimation here. Rather, we're going for a more broad effect, a more therapeutic effect of activating these neurons in different patterns to promote Hebbian plasticity. And so Sarah Mandela, who is a postdoc in the lab, she's now junior faculty in the Department of Rehabilitation Medicine, she's developed a really innovative way to chronically activate the spinal cord for optogenetics. It's a micro LED that's surgically implanted over a small laminectomy in the cervical lamina so that she can illuminate the dorsal surface of the spinal cord. She does an injection, a viral injection, to transduce some of those neurons to be light sensitive. Then she implants this electrode. And here's what it looks like, excuse me, LED. Here's what it looks like, just testing the LED. Every time you see a subtle blue flash down here on this circuit board, you should see flexion of the animal's left forepaw. And the electrical signal is passing through this cable, and then it's transduced into an optical signal as it leaves the LED. So this is just an example to show that Sarah can now robustly activate the cervical spinal cord optically for up to 6 weeks in these animals, do it during free behavior, do it while the animals are awake and reaching and using their forelimb in order to pair this activity with functional movements. And when she did that, she saw a very dramatic recovery of these animals treated with what we call optogenetic spinal stimulation. The blue line at the top are 3 animals, I believe, in this pilot study, recovering to near pre-injury levels, something we've never seen before with our control animals who hardly recover at all, or even our animals treated with electrical stimulation who look similar to these animals treated with light stimulation only, but without a virus to make their cells respond to light. Sarah's dug into the mechanisms behind this. She's looked at GAP43, which is a marker of axon growth or synaptogenesis. It's a marker of sprouting and plasticity. It's a little difficult to see here on this small slide, but the optogenetic animals, as indicated by the tall blue bar here, have about twice as much GAP43 after they've been stimulated by the light for 6 weeks. Light alone is not doing it, so it's not just a light or a heat effect. It has something to do with activating these cells optically. And so with Sarah's help, we've just been funded with an R01 for the next 4 years to really dig in and compare optical stimulation with electrical stimulation to see what it is that confers such a substantial advantage over the other things that we've tested. I didn't show the epidural stimulation here, but it's about similar to the green line. It's about half the recovery. So really exciting things ahead for Sarah and the rest of the team looking at optogenetic stimulation. Even if we don't translate optogenetics to humans, I think we'll learn a lot about the mechanisms by which these animals recover, and hopefully we can translate some of those mechanisms, for example, with a combination of pharmacology and electrical stimulation to human subjects. All right, I'm just going to come back to the outline slide to help us transition from our animal studies to our human trials, which we'll talk about for the rest of today. We'll highlight 2 studies, one focusing on upper extremity or hand function, and one focusing on standing and walking function. Both of these use spinal stimulation, but they use a less invasive variant. They use a non-invasive or transcutaneous spinal stimulation. Electrodes attached to the skin overlying the vertebra on the back of the neck. We'll add some electrodes in the lumbar cord when we focus on walking. But the innovation here is to use a very high frequency waveform overlaid on each stimulus pulse, a 10 kilohertz waveform, or 10,000 times per second, for each 1 millisecond wide stimulus pulse. And the reason to do this is to anesthetize or numb the C fibers, the pain fibers in the skin. These unmyelinated small nerves are more responsive to being blocked by high frequency stimulation. And so by delivering this high frequency pulse, we can anesthetize the skin, and it allows us to stimulate with 5 times more current than could otherwise be tolerated based on sensations on the skin. So we've tested this in over 20 people who tell us what feels the same on their skin, mostly people with sensory intact skin, a few people with sensory neuropathies, and they all reported on average that 5 times more current with the 10 kilohertz felt the same, qualitatively the same, as standard stimulation, FES or TENS stimulation without this carrier frequency. Another innovation we added to this study was to pair our transcutaneous stimulation with functional task practice, functional rehabilitation. So the idea that you do repetitive tasks with the physical therapist, with the occupational therapist, at the same time as we're stimulating the cervical spinal cord. We've had the opportunity to work with 7 participants in our study focusing on upper extremity or hand function. These are all people with cervical spinal cord injury, ranging in severity from Asia B, or motor complete, but sensory incomplete below the injury, to Asia D, but notably all our Asia D subjects have central cord syndrome, which I'm sure many of you are familiar with. The idea that there's a contusion in the cervical region of the spinal cord that actually affects the arms more than the legs. So despite the fact that they have an Asia D diagnosis, as you'll see from our first, actually both examples I'm going to show you today are individuals who are Asia D have very limited hand function at the beginning of the study. To some extent, can't grasp an object at all with their hands. But we've seen positive results in all of our participants. We'll highlight one of the Asia B subjects, 2 of the Asia D subjects, and a range of times post-injury, which we'll highlight as well, from as short as 1.5 years up to 12 years after injury. All right, this was our first participant. This was a collaboration with Dr. Christoph Hofstetter here in this department. Fatma Inanichi, who was a PhD student with us, now a postdoc at Harvard, and Soshi Samjima, who is was a PhD student, actually just defended a few weeks ago. I've got to update this slide. He's a physical therapist, now has his PhD, staying on as a postdoc. And we had the opportunity to work with this gentleman. His name's Joe. He's okay if we share that. He had his injury about 3 years before he joined the study. It was a cervical C3 Asia D cervical injury, and it left him with very limited hand function bilaterally, and you'll see that in the video. This was our very first subject, so we borrowed the stimulator and stimulated straight away and saw very substantial improvements in his hand function. This is the strength of 10 upper extremity muscles as measured by manual muscle tests. It's also part of this GRASP, or Graded Redefined Assessment of Somatosensation and Prehension, just a clinical and research test used to quantify hand function in spinal cord injury. So we saw big improvements in muscle strength. We also saw big improvements in function. The graph looks almost the same. We realized that we needed a control for physical therapy, and in all the subsequent subjects, we do the therapy alone before the stimulation. Maybe a subtle benefit of therapy alone, maybe a carryover effect. We're not sure with this subject. But the most important and exciting part of this graph is that for 3 months of follow-up, with no more stimulation, no more therapy, Joe maintained all of the strength and functional gains that he'd made during the study. So that's strong evidence for neuroplasticity. There's lasting benefits to this treatment that don't require the stimulator to be actively running or to have performed therapy within the last few days. All right, here's a video of Joe's hand function, first at baseline with the stimulator off. So you can appreciate here that his left hand, although he has some movement, he's unable to grasp a pinch between his fingers and thumb. This is a 9-hole peg test, and he can't get a hold of a single peg, although he's knocked a few out by getting them caught between his other digits. This is after 4 weeks of stimulation, and notably with the stimulator off. So this is the persistent benefit, or the neuroplastic benefit, at least immediately after the study. You see he can now reliably bring his finger and thumb together, move those pegs. It takes him a good minute and a quarter, so we fast forward in the middle, but he's able to do all the pegs with no mistakes, no drops. And then here, just the next day with the stimulator on, and this will give you a hint to the mechanisms. He's about twice as fast and twice as strong with the stimulator running. So we believe the stimulator is increasing the gain or the excitability of that 1A afferent motor neuron synapse, enabling this type of movement. But once people practice with the movement for many weeks, they don't require the stimulation in order to perform it. So we were excited about that, and we designed a more rigorous study to look at the effects of training alone done in the first month to the training of stimulation. And then because it was hard to decouple the carryover effects, we randomized people to either go back to training for a month and then more stimulation, or to continue stimulation and then training. So a bit of a crossover design here, but always at least 3 months of follow-up. And we're lucky to have 2 participants come back even for a 6-month data point, which I'll be able to show you today. First participant to go through this study was Asia B. So he had no movement below his wrist. He did have a little bit of a tenodesis grip, which makes the videos confusing to see. So we'll just show data on this case where we asked him to squeeze a pinch force, perform lateral pinch between his finger and his thumb on this transducer here. And at baseline and throughout a month of physical therapy alone, he could produce no force with either hand, right or left, shown by the 2 different colors, in that pinch force task. So he was effectively paralyzed, and he had not moved his fingers or his thumb in the 1.5 years since his injury. It was only after 2 to 3 weeks of stimulation combined with exactly the same therapy that he began to move for the first time since his injury, generate small but measurable pinch force on the sensor, just about a Newton or 2 here, so not yet functional, but another month of stimulation allowed him to approximately double that force again. Therapy alone didn't have any further benefit, but nicely here when he leaves, you can notice the point where he leaves the therapy session, and out till the end of a 6-month follow-up, he maintains this ability to move and this ability to pinch. And while this is weak force, right, less than 4 newtons might not be considered functional, combined with his tenodesis grip, right, which he has from preserved extensor muscles that are spared above the injury, he's able to learn to drive a self-modified car with hand controls, and also attempting to self-catheterize, which is a massive improvement in independence and dignity. I want to show you a couple videos of some of our other participants. This participant is Asia D, but 12 years post-injury, hasn't used his right hand functionally in the last 12 years. He's got a little movement, as you'll see in the video, but not strong enough to be functional. And this video is shot right before the stimulator was turned on, I think on the second day, and right after. And I'll narrate the first part, but then we'll listen to him speaking in the second part, because I think it makes a nice point about his impression of what the stimulator is doing to his function. All right, so this was done right at the end of training. He had been stimulated. We're asking him to lift these ping pong balls. These are very light, hollow plastic balls. You can see that while he's got a little bit of tenodesis grip, a tiny bit of function, he's not strong enough even to raise a ping pong ball high enough to place it in the yellow bucket. This is the first day of stimulation, so the very first time we had the stimulator turned on, and he had nearly immediate improvement in his ability to grasp and release, especially the releasing, which he actually had a lot of trouble with before as well. Sorry, the next video will be narrated with his reaction, but let me show you a little bit of data first, and then we'll look at his reaction. So this participant's name is John. He's okay if we share that. John came into the study with very little baseline function. He did improve a little bit, test to retest, proved a similar amount with a month of physical therapy. But it was only during these orange shaded regions where we applied the transcutaneous spinal stimulation along with the therapy where he made large gains in function. Training alone, no further gains. Another month of stimulation, sometime testing with the stimulator on versus off, on the orange box, off is the blue. So there's a little difference, especially in the early weeks. Often those differences converge after a period of time. And then again, 6 months of follow-up, if anything, steady or increasing function over those 6 months. And this is the strength score on the left, the same one we showed before. This is a score for prehension, or ability to bring the fingers and thumb together, such as the 9-hole peg test, putting coins in slots, keys in locks, nuts and bolts, a whole array of functional tests. Here, where he also improved almost exclusively during the stimulation period and then maintained at follow-up. All right, here's John's function right before and right after the stimulator was turned on for the second day. So we didn't get a chance to test these small wooden beads about the size and weight of a marble because he wasn't even able to do the larger ping pong balls shown in the background. This is with the stimulator off. So again, you can appreciate the weakness in his hands, a lot of effort to try to grasp these wooden beads, but unable to maintain pinch force to lift them into the bucket. This is just a few minutes later with the stimulator active.
Yes.
Oh yeah, that's crazy.
That's crazy.
Wow. Oops, I'll slow down a little bit. I'm acting like I'm a pro at this now. I'm just holding it and open it. Wow.
Helping, yes.
Oh
this
man.
is 45. Let's go with this one if you can
Oh
tolerate.
God, this is worth any kind of pain. This is amazing.
It's really amazing.
Wow. Like the other ones, like if I can get them, I'm like, they're just barely slipping out of my fingers. I can barely do it, but that's like a pinch.
So
That was really cool.
Yes. Yes.
It's even crushing. Wow, that is really, wow.
And so he continued to practice with his hands at home, but no more stimulation for 3 months of follow-up. And by the 3-month mark, he was certainly still able to grab those wooden beads and even grab tiny little friendship bracelets beads, things that I can barely pick up if they're left on the table. So exciting progress for John, and really exciting progress across all 6 subjects that were included in this more rigorous protocol. Each participant here in a different color. And you can see the effect of training alone, on average very small, just a couple points on these scales. And then that training combined with stimulation, everyone doing better at the end of stimulation for sure, very statistically different in pinch force, which I haven't shown you before. Just that tip to tip or lateral pinch force, grasp strength, and grasp prehension. So a couple other things to note with this study. One of the other ones was a very interesting reduction in spasticity. But I'll tell you right away that the spasticity is the one thing that's not lasting for many months after the stimulation. The spasticity appears to return a couple weeks after the stimulation is ended, which gives us some hints again to the mechanism. But I think you'll appreciate this video. This woman had such severe tone and spasticity in her fingers and thumb that although she had reasonable strength, she was unable to open her fingers and thumb to do anything functional. And you'll see her stretching the fingers and thumb to attempt to do this box and blocks task here at the beginning. So this is at baseline before any stimulation or training. She's about 2.5 years post-injury, C5 Asia C. A box and blocks task: pick up these 1-inch blocks, transport them over the barrier, drop them on the other side. Her fingers and thumb are essentially stuck together in a flexed posture, and her best solution is to scoop the block and kind of tip it over the edge. 12 days of training alone, and this is intensive functional task practice with the hands, what you'd get in a really intensive outpatient OT or PT setting. If anything, she's just sort of given up on the scooping technique. But just 3 days of stimulation, her tone, her spasticity has started to decrease enough that she can open her fingers and thumb and easily grab and transport these blocks. By 6 days of stimulation, she's gained enough control that she can precisely grab and release the blocks, right, to do a stacking task, something we hadn't been able to test before. And by this 18th day of stimulation, her tone was so reduced that we actually had to go out and buy larger blocks, 3-inch or 17-centimeter blocks, 7-centimeter blocks, that she could open her hand so far to grasp and releases. And remember, her fingers and thumb were completely stuck together before the study. So the unfortunate news about this is that after we discontinued stimulation, within about 2 to 4 weeks, that tone came right back. And that's an active area of research. And Christoph Hofstetter and I actually just submitted a DoD grant to look specifically at those mechanisms, both in our animal models and in our human subjects across the 2 labs. All right, I want to finish with this video partly because it's time for a musical interlude after all my talking, and also because it's so informative, I think, to listen to John describe his process for recovery. And so listen carefully to what he's saying about how he practiced, how he engaged in repetitive practice, because I'm not going to claim that this spinal stimulation is a magic bullet, whether it's epidural or transcutaneous spinal stimulation. I'm going to claim that the spinal stimulation enables people to move their hands when they can't, move their legs when they can't. We've seen that now across a number of sites. But it's that task practice, it's that rehabilitation that's happening while the stimulator is running that leads to the neuroplasticity. And I think John's narration of how he relearned to play the guitar 12 years after his injury, now 13 years, is a very interesting example of how the stimulator allowed him to practice, but he had to do the hard work of that practice. So just to put it in context, when he joined the study, we don't have a before video because we never thought he'd play the guitar again. Before he joined the study, his right hand didn't move at all, couldn't strum with his thumb, couldn't grip a guitar pick with his fingers. His left hand, all the fingers could move together, but not individually, and they were too weak to press the strings down to the frets. And you'll hear him kind of reflect on that recovery throughout this video. This was taken about halfway through the stimulation treatment period.
So you start like pull what you can use into it.
Yeah, what did your thumb do before on the right side?
Nothing.
Right, you couldn't hold
It just got in the way.
Yeah,
just like and I focus and work at it for an hour. You know, it's like we can strum it.
right.
And we can get that movement going. It's like I can't do it right now. So I'll just sit there for like a half hour and just like pulling it down.
That's why that particular movement's gotten so
That.
much better.
Just do it over and over and over until it starts to get a little better. Yeah. And that's how we even got here at all. Is I just would do this like just over and over until I could like, get it. It started like I couldn't even make the sounds. I couldn't push it down hard enough. And start to get it, and I started to do other ones, and pretty soon the chords start to come together. And I started to be able to play them on the neck. Which that's like all those fingers like working together.
And so John continued to practice for the 6 months after the study, continued to improve, and you'll see that here. Not particularly scientific, but interesting to understand that the stimulator is an enabler, and the practice and the repetition and the therapy are likely the things that are causing this long-term neuroplasticity. Just want to finish with a very short anecdote about some of the work we're doing with Rajiv Saigal to look at walking and to hopefully directly compare epidural and transcutaneous stimulation. We have an IRB already approved to do that. We've got multiple grants out trying to get the funding so we can implant people with epidural stimulator, not as cheap as you would imagine. In the meantime, we collected several subjects of transcutaneous stimulation, where we not only stimulated the cervical spinal cord, these are all people with cervical incomplete injury, so we're stimulating around the injury in the cervical cord, but we're adding electrodes around the lumbar enlargement, bony process T11 and L1, targeting that L2 central pattern generator. Same 10 kilohertz stimulation frequency. We use our body weight supported treadmill system. This is something over in the Amplifying Movement and Performance Lab, this wonderful collaborative space between rehabilitation medicine and engineering. So we can provide body weight support, can provide proportional movement of the treadmill based on their lean, can work on standing balance, we can work on stepping with and without body weight support, and we can work on overground locomotion either with a safety harness or in this case with a front-wheeled walker. This is our first participant. Again, Joe, if you recognized him from the videos, while he was in our hand study, he kept noting dramatic improvements in his leg function. So he came back for this study where he improved from needing full contact guard and leaning heavily on the front-wheeled walker to being able to take steps with standby assist or with the therapist so she just nearby for balance and using a single 4-point cane. He also improved the distance he could walk in 6 minutes. That was our clinical outcome for this, the 6-minute walk test. And here's stimulation, just to confuse you, is shown in blue, and training alone is shown in yellow. And so during periods of stimulation, he's making much greater progress. Actually, sorry, I got that backwards. Stimulation's in yellow, training alone is in blue. And so easier to see in the second participant, where there's an effective training, there's in both these individuals, but a steeper gain, a faster increase in function when the stimulation is combined. And certainly if we look at the averages across both participants, training alone has a modest effect, and the stimulation plus the training is about a 3 times greater effect. Almost all of those are maintained and well above the minimal clinically important difference. Just show a final video here and then we'll go to questions. This is our second participant who has an Asia D injury at C6 from about 4 years prior. He started stepping on the treadmill at baseline with about 20% body weight support and a therapist needing to move his right leg forward. He had weak ability to step with his right leg, and so that was the level of assistance that he required. We'll keep the body weight support the same, even though he was eventually able to walk without body weight support. Just for comparison, our goal was to push him to walk as fast as he could. So training alone, he can move about 0.1 meters per second faster. The first video is at 0.6 meters per second, and no longer required the assistance of the therapist. But as you'll see, after his 8 weeks of stimulation plus training, he's now able to walk about 3 times as fast. He's walking at 2 meters a second, again, with no assistance. We kept the body weight support the same just so you could compare. But again, he can walk over ground quite reliably at this point. So with that, I'll just summarize first the spinal stimulation results from our human trial and then just cap up the whole talk. And then happy to take questions for the rest of the hour. We've seen that non-invasive cervical spinal stimulation improves muscle strength and hand function. The benefits appear to initially require the stimulator, but once the people have practiced and trained, they maintain almost all the benefits even without stimulation active. Stimulation persists for up to 6 months, which is excellent. And the things I haven't had a chance to highlight yet, but are very functional and somewhat still a mystery at this point, is that almost everyone in the study experienced at least one autonomic improvement. It might have been improved bladder function, such as our first participant who no longer required intermittent catheterization after the study. 2 participants who improved bowel function, back to a regular daily bowel program. 1 participant, John, whose heart rate was severely bradycardic, which gave him orthostatic hypotension. 3 days, 4 days into stimulation, his heart rate came up to about 65 at rest, and it's been there ever since, which is a very interesting autonomic change. He also experienced sweating or diaphoresis, again, below his injury for the first time in 12 years, and some other just general sensation, being able to sense warm water on his skin and things like that. So there are very interesting, we'll call them off-target or anecdotal effects at this point. Almost everyone seems to get at least something. All right, recap of the talk. We started out way back with brain-controlled stimulation, first of muscles, showed that it worked in monkeys. 2 groups have taken it to clinical trial, seeing nice results other than that muscle fatigue. That muscle fatigue caused us to pivot and look at spinal stimulation, both intraspinal, epidural, and now transcutaneous, and even optogenetic. So trying to figure out what the benefits are of stimulating the spinal cord, what the mechanisms are that lead to these prolonged recoveries. And then finally finished off with this idea of therapeutic spinal stimulation, where we stimulate in order to enable people to perform their rehabilitation, but then are observing fantastic long-term effects that are definitely improving quality of life. So I want to thank the entire team that was involved in these studies, as well as all the funding agencies that have supported our work along the way across the bottom. And I'll thank you for your attention.